Good morning, everyone. My name is Alexandra Hamza from Manchester High School, and today I will be presenting on my research on the effect of Azospirillum lipiferum on the microbial recovery of corn soil after a moderate intensity fire. I would like to begin with a brief overview of my presentation today. First, I will begin with a background and introduction on fire and its effects on the microbial community, as well as a brief introduction into the process of inoculation. I will move on to my experimental objective, which of course affected my methodology, then my results, and any conclusions I could draw. There are 70,000 wildfires in the United States, and while we might only hear about a few, the soil microbial community keeps track of each one. After a high-intensity fire, it can take decades for the soil microbial community to restore itself. And while, of course, there might be an increased biomass of heterotrophic bacteria due to the soluble compounds that are released after fire, this is short-lived and often followed by a decline of biomass. And burned soil approaching pre-fire conditions must regain the same diversity of the pre-fire conditions, such as the right ratio of fungi to bacteria, as well as the right pH values, because burned soil often increases the pH of the soil due to ash. However, scientists have been using inoculation, which is placing beneficial bacteria in the soil, in order to shorten the span of time it takes for burned soil to approach pre-fire conditions. Cyanobacteria inoculation is one type of inoculation, and scientists have placed cyanobacteria into burned soil because it is able to produce a biocrust that can be inhabited by proliferating microorganisms. Arthobacter has also been used for inoculation. Arthobacter has been found to survive extreme environments, such as burned soil, and has found to increase plant growth after fire, and has been classified as a plant growth promoting organism. After learning about that, while there are 70,000 wildfires in the United States, 75% of those affect cropland and rangeland. I was led and shifted my vision to rangeland and cropland. And in this environment, Azospirillum lipiferum has been found as a successful inoculant in unburned soil. A lipiferum has been classified as a plant growth promoting organism due to its production of phytohormones, which aid in plant health and growth. A lipiferum is also a nitrogen-fixing bacterium. It can fix atmospheric nitrogen and makes fixed nitrogen available for plants and other bacteria. It has also had positive effects on corn plants in water-stressed conditions because it helps corn plants have an increased production of osmolytes, which are organic compounds that play a role in cell structure, especially during drought and water-stressed conditions. Knowing the positive effects A. lipiferum has on growth as well as the soil microbial community, I want to understand if these same positive effects also play a role in microbial recovery after fire. And I focus on diversity and abundance. And in this presentation, I will mostly focus on diversity. I also wanted to know if plant growth promoting effects of A. lipiferum are still in effect after fire. In my methodology today, I will focus on my fire process, the inoculation and planting process, and finally, the post-plant growth process. The fire process began at Foster Farm, where I collected soil from the roots of 11 corn plants. I collected a total of 10 gallons of soil, and the next day, the soil was burned. Half of the soil, about 5 gallons, was burned for a duration of 6 hours, and my goal temperature was 800 degrees Celsius because this is seen as the starting temperature for a wildfire. However, by 2 hours, the temperature I attained was 856 degrees Celsius. And of course, to get base levels for pH and microbial counts, plate count and pH was taken 3 days after the fire. 6 weeks after the fire is when I start inoculating as well as putting plants in. Burn and unburned soil were split up into 2 groups. I have burn uninoculated and burn inoculated, and the same was done with unburned soil. And half of the treatments were inoculated with a lipofurum, and plants were grown in each pot for a total of 16 pots. Pots and plants were placed in a greenhouse to mimic the month of June and the temperatures and the amount of light the plants would usually be given in the Yukon Floriculture greenhouse. And the image you see is about 2 weeks of growth of the corn plants. Finally, I would like to move on to post-plant growth, which is where I got the majority of my data that you'll see in my results today. Anything from above the white line is what I use to get my data from plant fresh and dry weight to get the total weights of the plant. And below this white line, this is where I got my total nitrogen values, soil for my plate counts, as well as pH. And for plant fresh and dry weight, as well as any other chemical or physical properties of the plant or soil, 16 out of 16 of the pots were used. However, for colony forming unit measurements as well as isolates, I only used 12 out of 16. So instead of 16 total pots, I used 3 pots from each treatment for a total of 12. And I chose 8 isolates from each treatment for a total of 32 isolates for 16S rRNA sequencing to get a gist of the diversity of the microbiome. And isolate sequences were analyzed using the nucleotide database from the National Center of Biotech and Information. Finally, I would like to move on to my results. Here, I will be talking about my nitrogen values, the microbial diversity, and a bit about abundance, and finally, plant growth. In my nitrogen values, I found that there was no true difference between burn uninoculated and burn inoculated. While, of course, there was a slight increase in the mean of the burn inoculated nitrogen, this was not significant enough. However, the true significance came out when I compared inoculated unburn versus inoculated burn, and I saw that fire did affect the nitrogen values. As you can see with this p-value, the unburned soil had a significantly greater nitrogen value than the burnt soil. Next, I would also like to focus on microbial diversity. On the x-axis, we see bacteria genera, and on the y-axis, we see number of bacteria that were found in the sequencing samples I had. And first, of course, I looked for similarities between a burned soil and an unburned soil in terms of the bacteria I found. And as you can see in the first column for bacillus, the unburned samples had mostly bacillus. And there's only one bacillus sample with my total burn samples for burn control. And burn inoculated, burn IN, as shown here, had no bacillus at all. Next, I looked for differences in gram-negative and gram-positive bacteria. Due to the thinner cell wall of gram-negative bacteria, it was important for me to see if A. lipofurum possibly played a role in increasing the population of gram-negative bacteria in the soil. What I found is that in burn inoculated, out of the 6 successful sequencing samples, 4 out of 6 of them were gram-negative and 2 of them were gram-positive. And in my burn control, out of the 7 successful PCR sequencing samples, only 3 of them were gram-negative, and the rest of the 4 of them were gram-positive. And this difference of 3 and 4 was not significant enough to say azospirillum played a true role in the proliferation of gram-negative bacteria after fire. Finally, I looked for differences in genera, and I did not find any true difference in genera between burn inoculated and burn control samples. I would also like to notice that in the previous slide, there are different total PCR sequence samples, and this is because some isolates that I selected were fungi. As you can see in the left, the organism that was circled was fungi, and this is why it is not shown in the PCR image as evidenced by the image on the left. I would like to move on to plant weight, and this is plant weight in unburned soil. In my x-axis, we see samples,
2 minutes.
and in my y-axis, we see plant weight. And of course, because Azospirillum lipiferum releases phytohormones for plants, I wanted to see if the same was found in burned soil. And I found that this was actually true. We see that uninoculated plants did not weigh 1 gram, and the least of them weighed at least like 0.75, while for inoculated plants, they reach 2 grams to 4 grams. The same pattern can also be found in unburned soil, where plant weight for inoculated plants did better and grew taller and had more success than uninoculated plants. And so what can we conclude about all of this? Well, I know that while burning did significantly decrease the amount of nitrogen that soil had, Alopeforum at this time of 1 month of growth had no significant change on total nitrogen. Also, no significant changes were present in the diversity of samples of burned soil due to Azospirillum lipiferum. While, however, we do know that the Bacillus genera was decreased after fire. And Alopeforum did play a significant role in plant growth in both burned and unburned soil. And this could possibly have an indirect effect on the microbial diversity over time. As plant growth increases, so will the root biomass. And as the root biomass increases, so will the production of root exudates, such as organic compounds and organic acids, that might affect the microbial population over time. And that concludes my presentation. However, I would like to thank the people at the Connecticut Agriculture Experience Station, including Dr. Blair Stephen and Jacqueline LaRue, and my mentor, Dr. Karen Kinsella, as well as my science research teacher, Wendy Turk, and the people at Yukon Blooms, the Yukon Floriculture Greenhouse, Nick Pettit and Shelley Duriger. Thank you.